Okay, so I think we can start with our class. So those who are new, so this is the second week discussion session or the live session of Fundamentals of Protein Chemistry, which is being taught by Professor Shagata Dasgupta of IIT Kharagpur. And I am Srijan Sen of IIT Kharagpur, and today we'll be taking the week 2 live session on Fundamentals of Protein Chemistry. Okay, so I am sharing my slides with you all. Okay, so like I go, firstly I will be discussing with the lecturers in very brief, and then we will be discussing a few question and answers in the later half. Okay. Okay, so I suppose my slides are visible to you all and I'm audible enough.
Yes, sir.
Okay, great. So yeah, so today's class, so last class we discussed the very basics of amino acids and peptide bonds. So from where we ended over there, we will start today's class. That is, today we will be discussing about protein architecture, that is building of proteins from these amino acids. So what are proteins? We all know these are biopolymers made up of 20 L alpha amino acids linked via peptide bonds. And there is a polypeptide backbone in a repeating sequence of NCC, NCC. So the NCC basically gets repeated where N is your amino group or the N terminus. Your middle C is the C alpha, to which you have the R group that is attached. Varying the R group, you get 20 different amino acids, and the other C is basically your carboxyl carbon. So these three atoms basically form the backbone of a polypeptide that later on folds to give us the protein molecules. Now, we discussed about the polypeptide chain or the peptide bond formation, where you have two amino acids, and via the dehydration reaction, that is, one molecule of water goes out, and you see that one peptide bond is formed. So one water molecule released is the driving force for the formation of a peptide bond between the two amino acids. So what did I say last day is that when we have a linear chain of amino acids, the number of amino acids that we have, n minus 1 water molecules will be released to give us a polypeptide chain. Or if we have a cyclic construct, on the other hand, then it will be n number of water molecules are released. That means n number of peptide bonds are actually getting formed. So from the example, you can see it where you have methionine, aspartic acid, leucine, and tyrosine. So this is a linear polypeptide, and three water molecules have been eliminated to give us three peptide bonds between methionine, aspartic acid, leucine, and leucine and tyrosine. Now, the characteristics of the peptide bond is that they are covalent. So covalent bonds are the strong bonds, and these peptide bonds have a partial double bond character. That means they neither have they are not as weak as a single bond or not even as strong as a double bond. Rather, the distance between the C and the N is basically 1.2 angstrom, that is somewhat in between 1.49 angstrom, that is for a single bond, and 1.27, that is for a double bond. The other important characteristic of a peptide bond is that it is rigid and planar, and it allows for the trans configuration. So the trans configuration is more prevailing because of the steric hindrance, to avoid any kind of steric hindrance during the folding of a protein. And the side chains, this also allows the trans conformation, also allows the free rotation of the peptide bonds on either side. Okay, so these side chains are very flexible. So now let us talk about the different types of bonds that we basically see in a protein molecule. We have covalent bonds, among which, so covalent are the strongest bonds. So we have peptide bond and the disulfide bond. So disulfide bonds are formed between, you all know, it is between the cysteine molecules. So the cysteines have SH group, so the hydrogen is lost from the two cysteines and there is a bond formation between the two sulfur groups. Okay, then comes the non-covalent bonds that are much weaker but relatively stronger and are very important in the folding of the protein or in the formation of the protein molecule. The first one being the hydrogen bond. The second one is the electrostatic interaction between the acidic and the basic residues, that is aspartic acid, lysine, or glutamic acid and arginine, somewhat like that. So these are the ionic bonds. And the hydrophobic interactions. So hydrophobic interactions are important. Why? Because they form the core of the protein, and the stable the core of the protein is, the more folded that structure would be, because it is not always that only the surface residues that form the hydrogen bonds play an important role. Rather, if you have a very stable core, they add to the even a better folding for the protein molecule in the end of the day. Okay, so these are the numerous types of bonds or interactions that take place during the folding of a protein or during the formation or generation of a protein molecule. Now if you go into the depth of these bonds, probably the disulfide bonds, you all know, is that it is found between the cysteine molecules, where there is an oxidation reaction where the hydrogen is lost from the two cysteines, and the disulfide bond is formed in an oxidized condition. So there is a redox reaction that is going on, and these disulfide bonds are very important, because if you get a very stable disulfide bond in the protein molecule, your protein is very stable. Okay, your protein is well folded, your protein's functionality is fantastic. But if there are multiple cysteines, what happens, if there can be multiple combinations of cysteine bonds that can be formed, and that might lead to wrong cysteine bonds being formed or misfolding of the protein, and that can lead to a loss of structure as well as function of the protein molecule. So correct cysteine bonds are very important to be formed. And this is a reverse reaction, because redox reactions are reversible reactions. So in a reduced condition, the cysteines remains as the single moieties. There is no disulfide bond between them. So when we run a gel, if you know that when we run SDS page gel, that is the sodium dodecyl sulfate polyacrylamide gel electrophoresis, what we do, we add BME to it, because BME is a reducing agent that allows the protein to denature entirely, also breaks the disulfide bonds, and that leads to the proper mobility of the proteins through the acrylamide gel, right? So one example of this cysteine bonds forming is the insulin, where you have chain A and chain B, and you see over here chain A has two cysteines that take part in the disulfide bond formation, that is intramolecular, and between chain A and chain B you have two intermolecular disulfide bond formation. If by mistake this cysteine goes and forms a bond with this cysteine, or this cysteine goes and forms a bond with this cysteine, it would lead to the loss of the insulin function actually. That is why proper formation of disulfide bonds are very important. The hydrogen bonds are much more simple, because we all know that it is the hydrogen from the electronegative, the hydrogen attached to the electronegative group interacts with the another electronegative, the electropositive, the electronegative group from the other portion. So you see you have an electronegative atom that is N minus, and you have hydrogen that is delta plus, and over here your C is delta plus and your O is delta minus. So by this we get the hydrogen bond formation. So hydrogen bond formation in simple words is the hydrogen has to be attached to the electronegative atom, and on the other hand the electronegative to which it actually interacts has to be attached to an atom that is in a delta positive condition. The ionic bond formation is basically occurs between the charged amino acids like lysine, aspartic acid, or a glutamic acid and arginine. So these electrostatic interactions are via the charge interactions or salt ion interactions, where the positive charge goes and climbs onto the negative charge of acidic amino acid, and then there is a bond formation or an interaction that is occurring that also leads to the stabilization of the protein structure. The I would call the most important interaction to be the hydrophobic interactions, because as I was saying, it is responsible for the formation of the core of the protein, because hydrophobic moieties are the ones that do not interact with the outside or the water portion, water molecules that are present outside in the surroundings. So these are the non-polar radicals that folds back inside, and the stable the core of a protein is depends on how well the protein is folded and how well the hydrophobic residues are arranged inside. So hydrophobic residues are responsible for forming the core. So what is the driving force for the formation of this hydrophobic interactions? It is that so when the peptide is or when the protein is in a linear form, you can see that both the non-polar and the polar residues are interacting with water, though the non-polar residues are not liking this condition. Okay, but still it has to, because it's in a unfolded state. But when it starts folding, what happens, the water molecules from the inside are basically kicked out of the core region where the non-polar molecule radicals goes inside. So they are removed, the water molecules, and that becomes the driving force for the non-polar residues to fold inside and form a stable hydrophobic core. So the stable the hydrophobic core is, the stable is, you can call the protein to be more stable. Okay, so you can see over here, this is an unfolded polypeptide and a folded conformation in an aqueous environment. The polar residues are outside, the hydrophobic core is basically inside. So in summary, the hydrophobic interactions and the van der Waals interactions are very critical for the folding of a protein. Apart from that, you have the hydrogen bonds and the disulfide, the ionic bonds that also contribute um, to a certain extent in the folding of the protein. And we all know about the covalent bonds, that is the disulfide bonds, that and the peptide bonds, that are the strongest bonds uh, that drives the protein to fold in a proper conformation. Just protein folding is not important. Folding of the protein in a proper conformation is very important, because the structure of the protein, the folding of the protein will also determine the function of that particular protein. So we do not want a protein that is folded but has no function at all. Then that makes no sense. That's just a wastage of time. What we need is a protein that is folded and is carrying out a certain function, like for example enzymes, catalysts and all. Okay. So the structure of the protein can be, you can categorize into four major structures, starting from the primary structure. Then it goes on to fold into a secondary structure, followed by the tertiary structures, and then you get a bigger domain where you have the quaternary structures. So the native protein structure is essential for the biological function of a protein. Loss of structure results in loss of biological function. So what is this primary structure? So the primary structure is the linear sequence of amino acids forming just the backbone of the protein. So it's just the backbone where the amino acids are joined via some peptide bonds and all, and maybe you have a few disulfide bonds that are linking it. So you have just the linear structure over there. There is no such folding or coiling and all. So it's just a simple linear structure over there where only the amino acids are present, joined by peptide bonds. So peptides can be dipeptide, tripeptide, tetrapeptide. Okay, and there are some oligopeptides as well as polypeptides that have even higher structure. So what is the difference between a polypeptide? Like it's not like that. What is the difference? Some, like, there might be some. These are basically interview questions or questions that basically people ask a lot of times. What's the difference between a peptide and a protein? So the peptide does not need to go and fold. So peptides can be as long as 50 to 80, even 100 amino acids. But if there is no folding, if it does not have a function, we cannot call it a protein. Okay, so it's like that. So peptides can be as long as tripeptides, even it can be as even longer than that, like oligopeptides and polypeptides, up to 80 to 100 amino acids as well. Now what happens when you actually alter or play around with the linear structure? So we know that each amino acid is coded by triplets, or so there are three base pairs, nucleotides, that actually determine the structure of a or that determine which amino acid will be chosen in which position. Like for example, if you want to go for a mutation or something like that, this is a natural mutation that occurs where one T gets exchanged with an A. One A is basically exchanged with a U, right? So you have the GAG is basically exchanged with GUG, and so GAG basically codes for glutamic acid, and GUG basically codes for a valine. So now when there is a change or a mutation that is occurring within a linear structure or a linear sequence of a protein, it actually hampers the function. And sometimes what happens, like for this case, from a normal beta globin, that's a normal hemoglobin cell, becomes a sickle cell, and that leads to sickle cell anemia, right? So one single mutation can basically change the entire function of a protein. Some mutations can be occurring naturally. These are some natural mutations, whereas some mutations are recombinantly introduced to increase the efficiency or the function of a particular protein, and that comes to under the protein engineering techniques and all, right? Now, so the protein and so the primary structure is basically very simple. You have a linear sequence and something like that. The secondary structure is basically the regular arrangements in space of adjacent amino acid residues in a polypeptide chain. So it's basically a folding or a conformation change within a particular sets of linear sequences. Like for example, if you have 100 residue long polypeptide chain, there can be the first 20 residues that is maybe forming that is folding in a different way. Then you have probably a gap. Then from residue number 35 to residue number 60, you get another structure, and then again at the end of the polypeptide for about 90 to 95 amino acids, you get a third structure. So this is just the folding of the different segments of the linear sequences that can happen depending on the arrangements of the amino acids, the arrangement of the sequences and all. And on the basis of that, there can be three types of secondary structures. One is the alpha helix. From the name we can understand there is a helix formation, a coiling. Then we have the beta sheet or the beta strands. So these are long segments like structures you see over here. And the last one is the beta turn, or we can call as the coiled regions. So coiled regions are very important to give the protein a little bit of space or some air to breathe. Okay, because the moment of alpha helix is ending, you cannot start with the protein cannot start with a beta sheet. It requires some gap regions and all to accommodate the coil regions or to accommodate a second folding, right? So what are alpha helices? So it's formed as a result of hydrogen bonds between the carbonyl oxygen and the amide hydrogen of the polypeptide chain, and does not depend on the side radicals. So you see over here the hydrogen of the nth amino acid is forming a hydrogen bond with the N plus fourth amino acid. So this is 1, 2, 3, and 4, right? So it's forming a hydrogen bond with the amino hydrogen of the N plus fourth amino acid. So the carbonyl carbon oxygen of each polypeptide chain is linked to a hydrogen bond with the amide hydrogen of the fourth amino acid towards the C terminus, and that leads to the coiling of this particular structure, right? We all know that every amino acid will have this carbonyl oxygen and the amino hydrogen. So alpha helices are very common in most protein structures, and the distance between the two between each of these coils. So this is an entire of the alpha helix. The folding of this coil is basically 3.6 residues, because there are 10 atoms that are accommodated over here, and it's like 36. So if you consider like 10 atoms are present over here, so 36 atoms can be accommodated. So that's 3.6 residues that is occurring per turn. So the pitch of an alpha helix is 3.6 residues per turn. What are the certain features of an alpha helix? So these are the most common because every protein will have, or every amino acid has a carbonyl oxygen and an amino hydrogen. So it's a very common and a stable configuration. So the spiral structure, that's the polypeptide bonds, form the backbone of the spiral. R groups of the amino acids remain outward of the spiral and does not actually affect it significantly in the coiling. And alpha helices are stabilized by hydrogen bonds. So hydrogen bonds are weak bonds, but it determines the entire or the total collective strength of all the hydrogen bonds is responsible for the stability of an alpha helix. Now, also there are certain amino acids that cannot be present in alpha helix, so we call them as helix breakers. So there are some amino acids that are helix formers or helix creators, but there are certain amino acids that cannot be present in an alpha helix, like proline and a glycine. Why is it so? Because the proline can attain a cis as well as a trans conformation. So the change of conformation of the proline can actually hamper the coiling of an alpha helix. Secondly, prolines have a very rigid structure. So when it has a very rigid structure, so what means it has to attain a rigid type of a folding. But alpha helix cannot always have a rigid folding. Maybe in one place it might fit in, but in most of the cases might be that rigid structure is actually creating a lot of difficulties, right? On the other hand, glycine is the smallest amino acid and it's the most flexible amino acid that's naturally occurring. So that higher degree of flexibility is actually affecting the formation of alpha helix. So where can we fit in these amino acids? These amino acids can be part of beta strands, little bit of beta strands, or they are most suitable in the coil regions. Why? Because sometimes there has to be a certain rigid turn. Okay, we call them as beta turns or turns that are actually very important in the contribution to the formation of a new secondary structure. So that place proline is very much feasible. At the same time, due to the flexibility of glycine, it allows the longer loop regions to be more flexible, allowing for the better folding of the protein. So yeah, we can accommodate these in the loop regions. Now also crowding of equally charged radicals like lysine, arginine, histidine, one after the other or at close proximity can also hamper the alpha helix, because there will be charge repulsion, the hydrogen bonds will not be formed, so the charge contribution acts more prominently than these hydrogen bonds, and it will affect the helix structure. The third one is the crowding of the bulk groups, like for longer side chains you have leucine, isoleucine, tyrosine, that leads to the spatial interference or steric inverse. And prevailing of amino acids with R groups are capable of forming the hydrogen bond. So we need to accommodate residues that also, where the R groups can also, if they are contributing significantly to the hydrogen bond formation, that will also lead to the stability of the alpha helix. And if we have certain harsh chemicals, that will also affect the alpha helix formation. Coming to the next one, that is the beta pleated sheets. It's more of a simple type where you have the anti-parallel beta sheets and the parallel beta sheets. So the parallel beta sheets run in a similar direction, where from the N terminus to the C terminus, the other beta sheet is also running in the N terminus to the C terminus, and you see that there is an elongated hydrogen bond and a distorted hydrogen bond as well. But on the other hand, the anti-parallel beta sheets are more prominent in proteins, because the distance between the oxygen and the electronegative hydrogen, electropositive hydrogen, is at a shorter distance, because we need these two atoms to be in close proximity as well. Now we cannot accommodate all of these in the parallel beta strands, but for an anti-parallel beta strands, where one is running from the N to the C terminus, the other one is running from the C to the N terminus, and that actually favors your protein folding and all. Now coming to the third one, that is the beta turns. Like I was mentioning, prolines and glycines are the favorable residues which we can place in the turns. Why? You see, for the two beta strands you need a beta turn. Why is this? turns important because proline, you see, have a very rigid structure, and it has a very rigid structure that allows only a strict folding over here. So you need this when there can be only 2 to 3 residues probably present over here. You keep a proline that will give you a short turn, and maybe a glycine that will add to a little bit of flexibility. You see that the antiparallel beta strand is forming fantastically. So it permits the change of direction of the peptide chain to get a folded structure, and beta turn loops allows for the protein compaction since the hydrophobic amino acids are on the inside of the protein, and polar residues tends to be on the outside, so beta turns out to be a more favorable one. Now the tertiary structures is basically a combination of the secondary structures. So there are multiple secondary structures that combine together to give you a three-dimensional shape. We call them as motifs or domains, depending on whether they are preserved over a long period of time or there is a new set of secondary structures that are actually forming. So it's a complete three-dimensional shape of a given protein, which we call as the conformation of the protein. It represents the spatial relationship of the different secondary structures to one another within a polypeptide chain, and how the secondary structures fold into three-dimensional form of the protein. So these tertiary structures can be like domains or motifs. So tertiary structure describes the relationship of different domains to one another within the protein. And so domains are basically a basic structural unit within a protein molecule. It's part of the protein, so combination of maybe 2 alpha helices, 1 beta strand, like an alpha beta alpha or a beta alpha beta. These are basically if they are conserved over a longer period of time, then we call them as domain. Otherwise, we just give them a name of motif. It's like whether they're historical or whether they're newly formed. On the basis of that, we have divided them into domains and motifs. So like a beta alpha beta, can be your like a motif, but like if this beta alpha beta domain is actually maintained for a long period of time, then we can call it a domain. So part of the protein that can fold into a stable structure independently, we call them as domains or motifs. Now different domains can possess different functionalities, right? Then what we have is proteins can have one or many domains depending on the protein size. At the same time, our polypeptide with 200 amino acids can contain more than 2 or more domains, and the domains are usually connected with the relatively flexible areas of the protein. Now these tertiary structures are stabilized by the interactions that I mentioned previously, the covalent bonds, ionic interactions, hydrogen bonds, hydrophobic interactions, polar residues interacting with the water. Then you have the alpha helices that have their individual hydrogen bond stability, as well as the beta pleated sheets. So these are the stabilizing interactions of the tertiary structure. I'm not going into the further details of these interactions. You all know, or I have discussed in details about these interactions, where you have the hydrophobic interactions between the non-polar alkyl and aromatic groups, hydrophilic interactions between polar and the surface water molecules, salt bridges between ionic interactions or the ionic interactions. Hydrogen bonds can occur between the secondary structures as well as the water molecules as well, the polar residues interacting, and the disulfide bonds. Now the quaternary structure is basically it's a bit complicated because there are multiple tertiary structures that get together to to form a quaternary structure. Like monomeric proteins are built of single polypeptide chains, whereas there are a few oligomeric proteins that are built of more than one polypeptide chains called the subunits or monomers. Why are they important? Because these monomers cannot function alone. They need their partner proteins or similar kinds of proteins that comes together like a monomer a particular protein might form a dimer, okay, or a tetramer, okay, or a hexamer, and this hexamer is basically functional. The monomer itself is not functional. So quaternary structures describes the joining of two or more polypeptide subunits. So two or more three-dimensional structures, tertiary structures, coming together give us the quaternary structure. The subunits have their own tertiary structures, and the bonds are non-covalent interactions between these tertiary structures. So it's like a primary folding into a secondary, secondary coming clubbing of two or three secondary structures gives us a domain or a motif, that is a tertiary structure, and the tertiary multiple tertiary structures gives us a multiple tertiary structures are called monomers. Individually they are called monomers, and these monomers come together to form a dimer, tetramer, or polymers. That is basically functional. So dissociation of the subunit, that is when it maybe is in a larger polymer form, and you break it down to a monomer, that can lead into the loss of function, right? So what is this basically? This is the Ramachandran plot. So this was done by Professor Ramachandran, and so this is important because there are different phi angles that determines the position of the secondary structures, right? Like an alpha, like a left-handed alpha helix, a right-handed alpha helix. You have the antiparallel beta sheets that are more towards the corner of the psi angles. Okay, so they are in the positive direction. So when you are plotting a Ramachandran plot, you plot psi versus phi, where you have phi in the x-axis and psi in the y-axis. You start from negative, so from the y-axis it's plus 180 to minus 180, that's the angle, and the phi is minus 180 to plus 180. You see that most of the antiparallel beta sheets are around this position, that is between plus 180 to around plus 90, and that is the psi angle, and for the phi angle it is between minus maybe 170 to around minus 60. Okay, so on the basis of this, and on the basis by generating a Ramachandran plot, we can tell about the structure or the composition of a particular protein that is not known to us. So if you have a new protein and we go on to calculate the Ramachandran plot by calculating the phi angles, we can tell the composition of the protein as to what are the secondary structures. Like over here we have the antiparallel beta sheets. The parallel beta sheets are also somewhat present in this region. So this region is contributed by the collagen triple helix, parallel beta sheets and antiparallel beta sheets. Around minus 60 to minus 75 to minus 50, around that we have a right-handed alpha helix, and the phi angle is minus 180 to around minus 60 or something like that. And then you have the left-handed alpha helix. So this Ramachandran plot is important for telling about the composition of the amino acid in terms of secondary structure, about the composition of the secondary structures. So as we were speaking about the quaternary structure, we have to speak about hemoglobin, because this hemoglobin is a globular protein consisting of 4 subunits, 2 alpha subunits and 2 beta subunits. You see the alpha chain, you have 2 alpha chains and 2 beta chains. In between this you have the Fe2 plus, where so each subunit contains a heme group for oxygen binding. So binding oxygen to one heme facilitates oxygen binding to the other subunits. So if you have a monomer, like only the alpha chain or only one alpha chain and beta chain, the oxygen will not go and bind. So binding of one oxygen to say probably the alpha chain will actually contribute to the binding of the oxygen to the other chains. So each of these chains have a heme group, and this heme group is responsible for binding of oxygen, and each chain, when one oxygen binds to one chain, this facilitates the binding of the other oxygen atoms to the other chains. And the primary structure is basically has no function. You need this quaternary structure, like I was telling. So these proteins, the quaternary proteins, work hand in hand. They cannot function all alone, right? Other one is the myoglobin. So it's present in our heart and skeletal muscles, striated muscles. So the kind of muscles that you consciously move from time to time, like your arms and legs. So myoglobin stores the energy in the oxygen in the muscles to facilitate the diffusion of oxygen from the blood to the mitochondria. That's the primary site of oxygen utilization in the cell. And the capacity of myoglobin to bind oxygen depends on the presence of the heme. So the heme group is a prosthetic helper group containing an iron atom. So you need to know a little bit about hemoglobin and myoglobin. That is why I did not talk much. Maybe whatever is important, I just spoke only about that. So protein structure, if you have already seen, it's hierarchical. That is from the primary structure, that is the sequence. It leads to the local folding of the primary structure, giving rise to alpha helix, beta strands, turns. That's the local folding or the formation of secondary structure. Then these secondary structure leads to long range of folding between the multiple regions. We get the tertiary structure, and these tertiary structures forms a multimer, and we get a quaternary structure. We can think till the quaternary structure, but there are obviously some supramolecular structures. And why do we need these supramolecular structures? It's very simple, because the monomer is not functional itself. You need more subunits of that particular moiety or that particular protein molecule to form a supramolecular structure that is important in carrying out the particular function. Okay. So why is so structure determines function. So if there is a loss of structure of the protein, it leads to disabilities in the function. But why is it so? Why is the function basically important? Because the protein function has a lot of roles to play apart from just the structure. It plays a vital role in the signaling molecules, cellular regulation pathways, catalysis like the enzymes, different cell movements like your catechols, nectins. Then there are cell transport proteins that allows the ion molecules to flow inside the cell or get out of the cell. So how can we determine the structure? There can be computational and experimental methods that can tell us about the structure and the function. Nowadays, finding the structure is simple after the discovery of AlphaFold, and there are so many updated versions of AlphaFold, and different bioinformatic tools are doing these AlphaFolds to tell us about the folding of the protein. But there is one problem. There are certain proteins that do not fold in orderness. We call them as disordered proteins. So AlphaFold cannot predict perfectly about the folding of disordered proteins. We need to keep that in mind. If your protein is unfolded, we can get only an idea about the ordered portion of the protein. The unfolded portion remains flexible, and you get some random structure for that unfolded region. So what determines the structure? Basically, it's the energy and the kinetimatics. So protein folding, the amino acid sequence of a protein determines the 3D fold. If you know the sequence of the protein, you can predict the structure of the particular protein depending on the type of tools that have developed. But previously they used to do manually, as the Anfinson's experiment, from where they had to introduce the cysteines and all, and from there they found out the folding of the protein and something like that. So what are some exceptions? All proteins can be denatured. Some proteins have multiple conformations that's difficult to predict, so you need to do some computational methods to get an idea about the stability and which conformation of the protein is more stable. And there are some proteins that get folding with the help of chaperones. You need chaperones or folding, some driving force to allow the folding of the protein. And the function of the protein is determined by its 3D fold. So can we predict the 3D fold of a protein given its amino acid sequence? We definitely can, depending on the type of tools that have developed nowadays. You can try it if you find maybe you go to the PDB structure of a protein, you just collect the amino acid sequence from the PDB protein data bank and put it in an AlphaFold. It's very simple to use. You have to just create an account and you have to just add the sequence. You get results within some seconds. So it's very simple nowadays. You do not have to do some rigorous experiments like an NMR spectroscopy or X-ray. We can do invest on the dynamics and something about the complex and all with the complicated experiments. So what is the Leventhal paradox? What is this Leventhal paradox? So for example, a protein has 100 amino acids and it can attain 3 different conformations. So it's like 3 different conformations that can be attained by 100 amino acids. So that's around 10 to the power 47 conformations. So depending on the fast scale motions of these conformations, it would take about 5 into 10 to the power 32 seconds. That's about 10 to the power 25 years. And that's a long time. So why are researchers even working on this? Because it is supposed to take 10 to the power 25 years to do it. So what is actually the driving force that is allowing for the folding of a protein and it's not allowing the other conformations not to form? Because it's a little bit of folding in the different regions that's getting stabilized. Every protein, every structure cannot get into 3 different conformations. So if a protein has 3 different conformations, we can assume that every peptide or every amino acid should have 3 different conformations, right? So if we agree on this, then but although we agree on this, but not every amino acid can attain 3 different structures. So that reduces the number, and ultimately we get the protein folding in probably a few minutes or hours, depending on the size of the protein and all, right? So that's the entire mystery about the Leventhal paradox. So the overview of energy, you need to know about the strength of the interactions or the contributions of the different interactions. Like hydrogen bonds have vary between the strength of an hydrogen bond varies between 3 to 7 kilocalorie per mole. The ionic bonds are like 10 kilocalories per mole. The hydrophobic interactions are a little bit less, like 1 to 2 kilocalories per mole. Van der Waals interaction 1 kilocalorie per mole, and the disulfide bonds are strongest, we all know, so 51 kilocalories per mole, right? Okay, so that's all about the lecture. If you have any questions, kindly ask me. If you have any questions anywhere, you can ask me. Then we will go into the solving of the questions, right? Maybe if I will give you a few minutes time to just recollect what has been taught, and then maybe we can move on to the questions. So no doubt on anything, you've understood everything. That's amazing then. Okay. See, the which bond is most strongest? It's basically the peptide bond. The backbone bonds are the most strongest. The peptide bonds. After that comes uh, about the disulfide bonds. Okay, because you cannot break the peptide bonds very easily. You need some very interesting enzymes as well as some so it's not very complicated breaking, because there are some enzymes that do it after identifying the series of amino acids. Okay, but you can easily add a reducing agent like a DTT or a BME or any kinds of reducing agents to break the disulfide bonds, but you cannot break peptide bonds that easily. Even disulfide bonds have been seen to dissociate in extreme heat, okay, but peptide bonds don't do that. Because when you run an SDS page, if you have run an SDS page, you know that you need to heat your samples to 100 degrees, and that often leads to, we had done experiments where it breaks the disulfide bonds, but the peptide bonds don't break. So the peptide bonds are the most stable among the covalent bonds, right? And next one, someone asked what is chaperones. So these are some protein molecules that allows the folding of the proteins. So these are also protein molecules. Okay, that's like when you are expressing proteins in bacteria. So these bacteria have some internal chaperones that allow for the folding of the protein, because when you work with bacterial proteins or something like that, the bacteria needs a driving force. If the protein folds on its own, that's fantastic, but in most of the cases you need some proteins or some driving forces to do that. Chaperones are like that. Anything else? Okay, even if you have any questions, maybe later on you can just ask me. I will be very happy to answer, or you can just drop me an email or something. um, I can also answer over there. That's not a problem. Okay, right, so shall I move on to the question and answers? So it's not chaperone, it's chaperons. It's CHA, and it starts like that. Chaperons. It's not. It does not give a shape. It's like that, but it allows to the folding. Okay, then let's move on to the question and answers. Right. So what I will be doing, first you people try to give me an answer, and then, so most of the questions are very simple. You can do it all alone, but if you are facing difficulties, I will just tell you if you people are going in the different direction. Okay. Okay. So maybe the first one. Which of the following bonds within a peptide backbone do not show free rotation? So first you people will give me an answer. Just give it a try. It's nothing like it's an exam or something. Just give it a try. You might be wrong, you might be right. Does not matter much. It will just boost your confidence. So question 1. Which of the following bonds within the peptide backbone do not show free rotation? What is the answer? Everyone try to give an answer. Okay. 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 So let's go one by one then. So the correct answer is B. Due to the conjugation of the nitrogen lone pair with the carbonyl group of the amide bond, because it has a partial double bond character, and we all know that double bond characters, even if they have a partial tendency of a double bond character, they don't go for a free rotation. So in C is correct, and that, so in C bond gets hindered, and the in C alpha, it does rotate in most of the cases, because we have seen that in C alpha is the phi angle, and phi angles vary for the secondary structures. So the carbonyl oxygen is the bond that does not rotate. So carbonyl oxygen and NC. Carbonyl oxygen, why? Because in the planar structure it tries to, because it also has a double bond character, as well as because of the partial double bond character, it's NC. The next one, please. Primary structure of a polypeptide is formed by, very easy. Okay, I've got one answer. Yes, It's very simple. It's a condensation reaction, removal of water, that leads to the structure of a polypeptide bond. Yeah, A is the correct answer. Good. Which bonds do not contribute in the primary structure? Okay, now this is interesting. Which bonds do not contribute in the primary structure? NC, disulfide, hydrogen bonding, salt bridge interaction. Okay. A, B, okay, no. I got just mixed up. So 2 and 4. So NC bond, that's the peptide bond, it's forms the backbone. So that does contribute. What about the disulfide bonds? Do they contribute? Yes, they indeed do contribute. I have shown in the primary structure that they can form, because the primary structure will not care about what amino acid is near to me. The cysteines will care about whether the medium is an oxidizing medium or a reducing medium, and based on that it can immediately form a disulfide bond over there. And the confusing options are basically B and D, right? Hydrogen bonding has no contribution in the primary structure. The moment there is a hydrogen bonding interaction, it actually goes into a secondary structure, right? So only the amide bond and the disulfide bonds contribute to the primary structure of the protein. So the correct answer is D. Hydrogen bonds and salt bridge interactions have no contribution. Okay, right. The next one. Molecular weight of a polypeptide can be determined by agarose gel, SDS page, MALDI. So MALDI is a mass spectrometry, or the elevation of boiling point. So molecular weight of the polypeptide can be determined precisely by. Okay, so I need to focus on that word, precisely by. So MALDI is matrix assisted laser deionization ionization mass spectrometry. You have a matrix over there, you add your sample to that matrix, you put it inside the spectrometer, the ions gets ejected out, and that is actually captured by the detector to tell you the mass. Okay, so how can you determine the mass of the molecular weight of the polypeptide? Yes, that is right. So SDS page cannot tell you about the exact mass of the protein, but MALDI can tell you it precisely. You get the exact mass over there, and mass spectrometers are fantastic in giving you the precise mass. But I have a question for you guys. Like, can you not tell about the size of a protein from the SDS page? Or if you want to determine the size of the protein from an SDS page, how can you do it? Anybody can tell me? If you want to know the size of the protein from an SDS page gel, how can you do it? Yes, we all know that we run ladders. We run different molecular weight ladders that we run alongside our sample names to tell us about a brief idea. But what about the precise? Can we tell it precisely? Though may not be precisely, but can we tell about the size? Okay, so I guess most of you have run an SDS page or seen an SDS page running. So you see that in the resolving gel or in the resolving layer alongside your standards that resolve according to the molecular weight, your proteins are also resolving. So how can you do it? You have to just calculate the length from the starting of the from the just the end of the stacking gel. So like this yellow line is the starting of the stacking gel. So from here your protein has run till say question 5. So you have to calculate the length of how much the protein has actually run depending on the band that you see over here, and your standards will run also like that. So you calculate the length of that, you take the logarithm of those lengths, you plot it against in log of molecular weight versus the length of the gel run, and from there you can also tell what is the size of your protein, because most proteins run uh, at the position where they are actually supposed to. So you need to do some analysis, but we can actually tell from the SDS gel where the band is actually coming, and just by determining the length and the log of the molecular weight of the standards as well as our protein, and by going through our reverse calculation, we can tell about the molecular weight of the protein just from the SDS page. Okay, next question. Secondary structure of a protein can be determined by absorption, CD, emission, single crystal extra diffraction. Yes, absolutely correct. CD spectroscopy will tell you about the secondary folding of the proteins. You get different dips, and determining on the dips, you can tell about whether it is a alpha helical ridge or a beta strand, or the percentage of alpha helices in the beta strands, and definitely from the extra diffraction you can tell about the secondary structure of a protein. Okay, alpha helices and beta sheets are primarily stabilized by hydrogen bonds, hydrophobic interactions, hydrogen bonds, electrostatic interactions, disulfide bonds, hydrogen bonds, and hydrogen bonds and hydrogen bonds. Which one? So alpha helices by which one, and the beta sheets by which one? Okay, I've got some answer. Maybe others can try. Excuse me. Okay, so most of you are telling disulfide bonds and hydrogen bonds. Disulfide bonds have nothing to do with alpha helices. Alpha helices are simply formed by hydrogen bonds between the innate amino acid carbonyl carbon, carbonyl oxygen, and the 8 plus fourth, that is the fourth amino acid, amino hydrogen. So there is no disulfide bond that is playing a role. So hydrogen bonds. And beta sheets, it's even simple. There has nothing to do with alpha helices, or it has nothing to do with any other interactions, because there are parallel and anti-parallel beta sheets. Both are driven by hydrogen bonds itself. So both are hydrogen bonds. So option D is correct. Now next one. What property makes myoglobin and hemoglobin different? Oxygen saturation profile at different pH, cooperative binding of oxygen, iron binding site, large segments of alpha helix with a spiral shape. Okay, most of you writing A. Yeah. So yes. So myoglobin shows higher oxygen saturation at lower pH, and hemoglobin shows uh, cooperative binding. So the oxygen saturation profile as well as the cooperative binding of oxygen will be different for a myoglobin and a hemoglobin. Which amino acids are most abundant in collagen? So which amino acids do you see the most in collagen? Okay, so what is basically a collagen? Collagens are proteins that have a triple helix structure. This structure is formed by the three polypeptide chains that are intertwined between them, and each of these sequences, or the driving most of the sequences, is a repetition of glycine, proline, and some other amino acids, like the different amino acids not naturally occurring, like the hydroxyproline. And this uh, you can tell this arrangement of proteins are important for the formation of the triple helix, because our collagen is basically important for its strength as well as flexibility, and prolines and glycines are the important residues, or play an important residues in the structure and the stability and the flexibility. Okay, so prolines and glycines are most abundant. Now the interacting surface of a membrane protein with the membrane is, okay, so the interacting surface is hydrophilic, hydrophobic, amphiphilic, or it's unpredictable. So it's telling about the interacting surface. So the interacting surface is a PLP bilayer, right? So it is hydrophobic in nature. Hence, to interact with the membrane, proteins have to be hydrophobic to undergo hydrophobic interaction. Correct. Which of the following does not affect the stability of alpha helix? Hydrogen bonding, branching at C alpha position, presence of alanine and serine residues, interaction between R groups spaced 3 residues apart. Does not affect the stability of an alpha helix. Yes, that is correct. Branching at the C alpha position can lead to steric hindrance, and that plays an important role in the affecting the stability. Which secondary structure has a strong unidirectional dipole moment? Alpha helix, beta strand, parallel beta strand, antiparallel beta strand, or a term? Unidirectional dipole moment. Alpha helix is the correct answer because why alpha helix is the correct answer? Because it folds in a manner where all the N terminals are aligned on the top and all the C terminals are aligned on the bottom. So it generates a unidirectional dipole moment where there is a delta positive charge towards the upper portion and there is a constant negative charge at the lower portion due to the carbonyl oxygen, and the positive the delta positive for the N terminus is due to the contributing delta positive charges of the collaborative delta positive charges of the nitrogens, right? Okay, question number 10. So it says which of the following does not affect the stability? So the answer is branching at C alpha. Why? Because the more the branching at C alpha, it means it is hampering the hydrogen bonding or the folding in a particular conformation of the neighboring atoms that can lead to steric hindrance and hampering of the alpha helix formation. Okay. So more branching can lead to a problem in the alpha helix formation. Question 12. The factors responsible for the adoption of a specific protein conformation in an aqueous solution are formation of maximum number of hydrophilic interactions, yes, it says which does not affect the stability. So the branching of the alpha carbons does affect the stability. That is what I'm trying to tell. Hydrogen bonding does not affect. Oh. Right, right. Yes. So there is a mistake. Yeah. Which of the following does not affect the stability of the alpha helix will be basically hydrogen bonding. It does not affect the stability of the alpha helix. It's not branching of the C alpha. Yeah, thanks for correcting. Yeah, that is right. And if it's a multiple choice, then you can go for A, C, uh, probably D. Yeah, right. A, C, D can be correct. Okay. Yeah, that is right. Okay, formation of the maximum number of hydrophilic interactions, minimization of entropy by the formation of water solvent cell around the protein, placement of hydrophobic amino acid residues within the interior of the protein, placement of polar amino acid residues around the exterior of the protein, or formation of maximum number of hydrogen bonds. So what are the factors that are important for the specific protein conformation in the aqueous solution? So what is important? Let's go one by one. Formation of maximum number of hydrophilic interactions, that is correct as well. Minimization of entropy by the formation of water solvent cells around the protein. Placement of hydrophobic amino acid residues within the interior of the protein, that is also correct. So we do not have 1 and 3 over here. Let's see. Placement of polar amino acid residues around the exterior of the protein, that is also contributing. Formation of maximum number of hydrogen bonds. So probably the options that are given over here are not actually very correct, because according to me all the 5 options are actually correct, right? Because the number of hydrophilic interactions are important, and more number of polar residues increase the hydrophilic interactions. The hydrophobic core is controlled by the hydrophobic amino acid residues in the interior. Maximum number of hydrogen bonds will actually lead to the stability of the protein, better stability, and maximization of entropy of the water. Okay, minimization of the entropy is also required around the protein. So all the 5 options are correct over here. Okay, now a hypothetical helix has 5.6 residues per turn with 5.2 angstrom pitch. What is the rotational angle per residue of that helix? So there is a helix that has 5.6 residues per turn and the pitch is 5.2. So what is the rotational angle per residue? So how do you do this? So what is the total rotation that can actually occur? That is 360 degrees, right? And how many residues are there per turn? 5.6 residues. So what is the rotational angle per residue of that helix? 360 by 5.6 residues. So how much is it coming? So 6, that is probably around 64.3, or I'm not sure. So 360 by 5.6, that is 64.3. So B is the correct option. And the last one is dihedral angles phi and psi for beta sheet are respectively. What is the phi and the psi of a beta sheet? Positive, negative, negative, positive, positive, negative, positive, positive, negative, positive, positive, negative, or negative, negative. So what will be the dihedral angle of a beta sheet? We just discussed about it. So it is beta sheets are present at the top corner. So what does the top corner signify? Minus 180 to, so top left corner. So it means that psi is negative, and what about psi? Sorry, what about phi? Psi is negative. What about phi? That is also negative. So option D is correct. Right? So you can see from here. See beta sheets over here. Okay, so this is positive, sorry. So the psi is actually positive and the phi is actually negative. So Phi is negative and Chi is positive. So option B. Option B is the correct option. Okay. So that's all then. Um, if you have any questions, you can take. So how did I do question number 13? So it is telling there is a helix with 5.6 residues per turn with 5.2 angstrom pitch. So here the pitch is just given as just for as a build a confusion, but the total turn that can occur is a 360 degrees. So 360 degrees divided by number of residues per turn, that is 5.6. So 360 by 5.6, that is 64.3. Okay. Anything else? I am done from my side. If you have anything to say, you can tell me. So we can end the class then, right? Okay, good. So anyway, I will be sharing the slides and the recorded video lecture with you all. If you have any problems, you can just mail me in my mail ID. I will be happy to answer. Or you can ask me in the next class. That is also fine for me. Okay. Thank you then. Again, we will meet in the next Friday at 6 o'clock. Okay. Good. Thank you very much for joining.